Thanks for tuning into this VUMEDI short podcast about mantle cell lymphoma and the recent FDA approval of the Acalabrutinib plus bendamustine rituximab regimen from the ECHO trial. So my name's Chan Chia. I'm a hematologist at Sir Charles Gardner Hospital in Perth, Western Australia, and I was an investigator on this study. As a way of background, patients with mantle cell lymphoma traditionally have been treated with chemoimmunotherapy historically, with the main division in treatment being whether or not patients were suitable for high-dose therapy, autologous transplantation. And in the older age group, so that's been considered patients 65 years or older, or those with comorbidities that make them ineligible for high-dose therapy, the standard of care, or one of the standards of care, has been bendamustine and rituximab. It's reasonably effective, results in a median PFS of somewhere between 4 and 5 years, is reasonably well tolerated. There was a prior study with a very similar design called SHINE, in which patients were randomized to BR plus or minus ibrutinib, which is of course the first-in-class BTK inhibitor, and the ibrutinib was given continuously until disease progression or unacceptable toxicity. Now, there was a progression-free survival advantage on that trial, but the overall survival was not improved at all. In fact, the trend there was a negative trend towards overall survival for patients who had the experimental arm in which ibrutinib was used. And so as a consequence, the SHINE regimen never actually was approved by the FDA. Acalabrutinib is a second-generation covalent BTK inhibitor, which has a favourable side effect profile relative to ibrutinib. Because it's cleaner on BTK, there is less off-target toxicity, lower rates of things like atrial fibrillation, and as a consequence, the ECHO study was performed to evaluate the benefit of the addition of acalabrutinib to the bendamustine rituximab regimen in patients with previously untreated mantle cell lymphoma, age 65 or older, and otherwise unsuitable for autologous stem cell transplantation. You can see the schema for the study summarized here. Patients were randomized 1-to-1 to either BR plus placebo or BR plus acalabrutinib. The placebo arm is illustrated in orange on subsequent slides, and the experimental arm containing acalabrutinib is in blue on this diagram and on subsequent curves, so just remember those numbers. So inclusion criteria in the purple box to the left, essentially reasonable organ function, performance status, and MCL, in need of treatment, 65 or older, and not eligible for stem cell transplantation. Stratification by simplified MIPI score and geographic region. Important to note, enrolment took place between 2017 and 2023 at 195 global sites, and this time period obviously does include the COVID pandemic, and this did have some effects, which we'll sort of cover a bit later. The primary endpoint was progression-free survival, with key secondary endpoints overall response rate, overall survival, and safety. The demographics and baseline characteristics of patients are summarized here. Approximately 300 patients in each arm. They were well balanced for baseline characteristics. A few things to highlight: male predominance, as you would expect, median age of 71, with about 1/3 of patients over 75 years of age, so an elderly population, predominantly male, as you would expect for this entity. 13% approximately of patients with blastoid or pleomorphic histology, and about 25% of patients with high-risk simplified mantle cell IPI. The TP53 status was actually unknown in the majority of patients, but among those where it was, among the patients overall, about 8 to 10% of patients had TP53 mutations, and around 30% were known to be unmutated for TP53, with the remainder unknown. Higher proportion of patients had Ki67 status evaluated by immunohistochemistry, and you can see that patients with higher risk, in other words, Ki67 of 30% or greater, comprised approximately 46 to 50% of patients in the acalabrutinib and placebo arms, respectively. In terms of overall response rate, there was a trend toward higher overall response rate in the experimental arm, and indeed the complete response rate was higher for patients who received acalabrutinib, 66 versus 53%. The primary endpoint of this study, however, was progression-free survival, and you would argue that that's clinically more meaningful for patients with mantle cell lymphoma. And there was a significant benefit in terms of PFS for patients who were randomized to acalabrutinib. The median PFS was improved by 17 months, which was a hazard ratio of 0.73. So that means a 27% reduction in the risk of disease progression or death. One important point to highlight in terms of difference in design between ECHO and SHINE is that within ECHO's study, patients who experienced disease progression on the experimental arm were able to cross over to acalabrutinib on study. And as a result, a higher proportion of patients who experienced disease progression on the placebo arm subsequently received a covalent BTK inhibitor as their next treatment, which is higher than the proportion of patients on SHINE who did so. I think from memory, it was about 30% on SHINE, close to 70% on ECHO. And what this means is that in terms of a sequencing question, although this study was not specifically designed to ask the question of whether sequencing treatment, so in other words, BR with continuous acalabrutinib versus BR followed by a covalent BTK inhibitor at the time of disease progression, it wasn't answering a PFS2 question, but we can make some observations about the impact on overall survival from the fact that 69% of patients receive BTK as their next line of therapy. So this is relevant when we consider the overall survival curve. And although there was no significant difference here, the hazard ratio here is 0.86 with a p-value that's 0.27, the trend is toward a benefit for acalabrutinib, even in the presence of crossover, and certainly no suggestion that there were an excess of deaths in patients who received acalabrutinib in terms of increase in toxicity, which was the case in SHINE. So in terms of safety, the majority of patients had an adverse event on study, and 12% and 10% were grade 5 in the experimental and placebo arms respectively. When considering adverse events of particular interest in patients treated with a covalent BTK inhibitor such as AF, hypertension, bleeding and infections, you can see that AF rates were only marginally increased in the acalabrutinib arm versus placebo. So any grade 6.6 versus 4%, grade 3 or higher, 4 versus 2%. Hypertension was similar, if anything numerically lower in the acalabrutinib arm. Major bleeding was similar, if anything lower on the acalabrutinib arm, and infections perhaps marginally increased, with no difference in second primary malignancies, and that's despite a longer median treatment exposure on the acalabrutinib arm. Coming back to deaths on study, about 32% and 35% respectively of patients in the acala and placebo arms died on study. There was a higher number of deaths due to disease progression on the placebo arm, and adverse event-related deaths were not significantly increased in the acalabrutinib arm. COVID-19 is worth mentioning, and there was an increase in grade 3 or higher and grade 5 COVID-19 adverse events, and there was a higher number of patients discontinuing due to acalabrutinib or placebo in the due to COVID-19 as well. So the important thing to recognize is that because this study was took part during the pandemic, it's unclear whether this trend would still be seen in patients who were vaccinated and had access to antiviral therapy. If we consider again the PFS and overall survival with a COVID-19 pre-specified sensitivity analysis, you can see that with COVID-19 deaths censored, if anything, the trend toward PFS and overall survival benefit is greater. The hazard ratio with the COVID-19 censoring is 0.64, which is a lower hazard ratio than the full analysis population. And again, similar trends seen for overall survival, 0.75 versus 0.86. This did not reach significance. However, the trend certainly increased there. So I think we can conclude from the ECHO trial that the addition of acalabrutinib, given continuously until disease progression or unacceptable toxicity, certainly resulted in PFS benefit and trend toward overall survival benefit when used as a continuous therapy. The censoring for COVID-19 deaths certainly seemed to help things. The safety profile seemed to be better than the ibrutinib plus BR combination. And as a consequence, I think this was sufficient evidence for the FDA to approve this as a first-line therapy for patients with mantle cell lymphoma. There was a follow-up abstract presented at the ASH meeting, which looked at a subgroup analysis, and I can share those the forest plot with you there. Right. So this was the subgroup analysis recently presented at ASH, and the key points here I want to focus on are firstly the subgroup analysis for patients with various subgroups, and this is the key figure here. Overall, this is a PFS forest plot showing the impact of various subgroups. To the left favours acalabrutinib and BR, to the right favours placebo and BR. You can see that across the subgroups analysed, in terms of sex, age, geographic region, baseline ECOG, tumour bulk, MCL subtype, TP53, mutation, Ki67 stratified at 30%, there was a consistent trend toward benefit for the acalabrutinib BR arm. Not all of the subgroups, of course, were significant because the study overall was not powered to detect PFS differences in each individual subgroup. But the point is that the trend was in a similar direction in all patients. You can see, importantly, back on the benefit in terms of COVID-19, you can see that the patients who were vaccinated, which was a higher proportion of patients, there was a greater trend toward PFS benefit, which probably is a direct impact of the excess COVID-19 deaths in patients who were unvaccinated. So that should give, some consolation to the fact that if this study were redone in the current era with higher rates of vaccination, access to antivirals, it's likely that the PFS benefit may be greater. And importantly, patients with high-risk features such as TP53 mutation and high Ki67 blastoid pleomorphic histology, there was, a trend towards benefit for the high-risk subgroups, although clearly some of these subgroups were small numbers of patients, and therefore we can't sort of overinterpret it, but there was no suggestion that there was a lack of benefit there. There was some data also presented about MRD conversion, and this was measured by clonoSEQ in the peripheral blood, and there was a higher rate of conversion of MRD of MRD positivity to MRD negativity in patients on the acalabrutinib arm, 37% versus 20%, recognizing that this is relatively low numbers of patients, and a lower proportion of patients who went from being MRD negative to MRD positive in those who received acalabrutinib versus those patients who received placebo in addition to BR. So we've got additional analyses here suggesting that high-risk subgroups look like they have a similar benefit compared with the overall population, and also that there are higher rates of conversion to MRD negativity in patients who receive acalabrutinib as well. So I think in summary, we've got now access to a first-line regimen containing a covalent BTK inhibitor in patients with transplant-ineligible mantle cell lymphoma. We saw a lot of data at the ASH meeting about this shift towards the use of covalent BTK inhibitors in first line, and I think that's increasingly something that we'll see, potentially even in non-chemotherapy-based regimens. We saw a number of those presented at ASH as well. So the future for patients with mantle cell lymphoma looks increasingly bright. Thanks for your attention.